Thanks for coming. I'm just going to briefly introduce the speakers. Mr. Guilherme Saturnino is a PhD student at the Danish Research Center for Magnetic Resonance and the Technical University of Denmark. His main interests are developing tools for simulating electromagnetic fields from transcranial brain stimulation and leverage those tools for advancing the TBS. He has worked on SimNIBS, an open source software package for simulating electric fields caused by TBS since 2013, where he has developed new methods of electrode modeling and uncertainty quantification, post-processing, FEM simulation, and optimization. And Mr. Oula Puonti is a postdoc at the Danish Research Center for Magnetic Resonance and the Technical University of Denmark. His research focuses on developing computational methods for analyzing medical images based on probabilistic modeling and applying these tools to study the neuroanatomy in healthy and pathological brains. Currently, he is working on developing a robust and accurate segmentation approach for creating realistic head models used for TBS simulation in SimNIBS.
All right, thanks for the introduction, and thanks for giving me and Guilherme a chance to come here and talk about SimNIBS, which is an open source tool for individual. Yes. Sorry? Oh, the mic is not on.
It's not working. No, yeah, it's working.
It is. Should I be like here? Can you hear me now? All right. Okay. I hope. Okay, I'll try. I'll try to speak louder.
So the microphones should be.
Okay.
I mean, it's working, but like not so.
Yeah, just not capturing.
It's not capturing. Should I be here? I can do it like this. It's okay. It's better like this. Okay. Yeah, no, it's all right.
Can you turn it up a little bit?
I think there is at the maximum.
Is it the maximum? Okay. Well, I'll stay close to it. Yeah. Okay. All right. So anyway, yes, thanks. So we're gonna present SimNIBS, which is an open source tool for individualized simulation of the electric fields induced by transcranial electric or transcranial magnetic stimulation. And SimNIBS is being developed at the Danish Research Center for Magnetic Resonance, DRSMR, and also at the Technical University of Denmark. Just tell me if you don't hear me. So here's an overview of the talk. So I'll start with a short motivation on why it makes sense to use these individualized models. Maybe this is familiar to some of you already. Then I'll show the overview of the SimNIBS pipeline in general, what the different parts are. And then we'll look into the different parts in more detail. So I'm going to talk about anatomical modeling, specifically segmentations of head tissues from MRI scans. And I'll talk a little bit about how segmentation accuracy affects the E-field simulations, how MRI scan parameters affect the segmentations, and through there also the E-field simulations. And then show some initial results on how the fields look like if you actually include multiple tissues into the head model. And the second part, we'll switch speakers, and Guilherme is going to talk about uncertainty quantification, so how to map uncertainties in conductivity estimates in the tissues to actual field simulations, and also about how to optimize the electrode positions for transcranial electric stimulation. So this motivating example comes from TES, transcranial electric stimulation. So in typical sort of application, you use this sort of one-size-fits-all stimulation protocol where you use these large pad electrodes and you place them into sort of approximately the same place in all your subjects in the study. And this doesn't account for anatomical differences between individuals. And what you usually end up with is these very distributed electric fields, which might be a reason for the quite widely reported inter-individual variability in responses to electric stimulation. So what we would like to have instead is to start by defining a target on the cortex and then optimize the electrode positions and the input current so that we hit the target with high intensity electric field strength and minimize the electric field outside of the target. And this sort of doing this optimization in a way where you actually also account for individual anatomical differences like scalp thickness or amount of CSF So if we do it in this way, we can actually make sure that the dosing is the same across all subjects. So we're actually stimulating with the same intensity and in the same location. And this way we can then start understanding if stimulating a specific brain area with specific intensity actually gives us a response. For TMS, the anatomical features that affect the field in the brain are mostly the coil to cortex distance, the individual gyrification pattern, amount of CSF, maybe the inner border of the skull. But especially if you move into these sort of multi-coil systems, then this individualized modeling becomes more important for TMS as well. So this is sort of the schematic of how the SimNIBS pipeline looks like, or maybe any pipeline for E-field modeling, individualized E-field modeling. So we start out with the subject MR scan, typically at T1. We would also like to have T2s. And the first step is to do segmentation. Usually you have 5 different tissue classes. So white matter, gray matter, CSF, bone, and soft tissues. Sometimes the bone is separated into the spongy part and compact part, but typically you have 5 tissues here. Once we have that volume segmentation, we generate a finite element mesh from that. And here we can then do our simulations for different electrode positions, input currents, coil positions, or different coil models. Or we can start by specifying a target in the mesh and then optimizing the electrode position and input currents so as to maximally hit the target. So as I mentioned before, I'm going to cover the first part, and then Guilherme is going to do the second part. So once we started working on this automated segmentation of the head tissues from MR, we realized that there's not really many datasets out there where we have high quality segmentations of multiple tissue classes on multiple subjects. So what we started out with was by acquiring such a dataset. So here we have, in this dataset, we have 20 subjects. The dataset was split so that we have 10 European subjects and 10 Asian subjects. And those groups were again split so that we have 5 male and 5 female subjects. And the age range was from 20-year-olds to 50-year-olds. So it's a fairly diverse dataset demographically. And the subjects went through quite a lot of head scanning. So we have multiple different MR contrasts, T1, T2, M. Dixon scans for water and fat, also a venogram, and we also have a CT to see the bone. So the idea here was really to sort of do a lot of different contrasts to see all the tissues that we're interested in so that those can be segmented out accurately. And the segmentations were done by our collaborators, the ETIS Foundation in Switzerland. And here you can see the cortical or the surface reconstructions of the different tissues for one subject. So we have the standard white, gray CSF, but we also have, for example, the visual nerve, the spine, the eyes, the eye muscles, major veins and arteries in the head. We have the bone, which is now split into the compact and spongy part. And then we have a couple of different smaller tissues, and the soft tissues are sort of lumped together in a single tissue class. So in total, we have 14 different tissues in these segmentations. So given those sort of manual or semi-automated segmentations, we then developed a segmentation algorithm based on this. And this is based on some of our previous work in whole brain segmentation, which is now also being tested in the Free Surfer group. And this sort of algorithm has 2 parts. We have a probabilistic atlas of the head anatomy, and this is now a mesh-based atlas. This should not be sort of mixed up with a FEM mesh. This is not a FEM mesh, it's just a probabilistic encoding of the anatomy. And the second part is an intensity model where we then sort of model how the different tissue classes look like in the MR scans. For example, how is the intensity distribution in the white matter. And these 2 parts of the model are fitted onto the input scan. So we deform the atlas so that it matches the anatomy of the input. And we also fit the intensity distribution so that we then know what, for example, white matter looks like in the input scan. And the idea here was to be sequence adaptive. So if we get scans from different scanners or centers or sequences, we can sort of do the segmentation without retraining the model just by learning these parameters. The other aim was to try to be robust. So if we also get clinical scans, which usually have a lower resolution and movement-related artifacts, we should be able to sort of get reasonable segmentations out of those as well. And here you can see an example of a T1 weighted input and how the segmentation looks like. And now we have these different classes. For example, the spongy bone is in this reddish color, compact bone is in more brownish, and we also have the vein in blue. So you have all these different tissue classes here. So once we had that segmentation algorithm developed, we wanted to, and now we call this thing CHARM, because you have to have a catchy acronym. So we wanted to compare it to other tools, open source tools for doing individualized simulations, and we compared it to ROAST and to SimNIBS, the current segmentation approach in SimNIBS. And both these ROAST and SimNIBS segmentation approaches are SPM based. And what I'm showing here is the die scores for these 5 standard tissue classes on 20 subjects. And the filled boxes correspond to a case where we only have a T1 weighted scan as an input. And the striped boxes correspond to a case where we also have a T2 weighted scan. So what you can see here is, if you look at the CSF and bone classes, is that if you include a T2 weighted scan, the segmentation accuracy increases somewhat, and this is because CSF lights up in the T2 weighted scans, and you can segment the bone CSF border more accurately. This new tool CHARM does quite well already with a T1 weighted scan. If you include a T2 weighted scan, you get a little bit of an increase in the CSF and bone scores, but it doesn't look like much, but I'll show some results for E fields, and actually these differences can affect the fields by quite a lot still. So the reason we care about segmentation accuracy is because segmentation differences result in E field differences in the simulations. So here are 2 subjects, example subjects where the first segmentation is from the SPM-based segmentation method in SimNIBS, and the second one is now from this new tool, CHARM. And what you can see, if you look at the current segmentation method in SimNIBS, is that you get this under-segmentation of the skull in many places. And for the first subject, this thinning of the skull happens to be right under the stimulating electrode in this TES configuration. And this is why you get these very high field intensities on the cortex. Whereas if you actually segment the skull correctly, these field peaks are much reduced. Same goes for the other subject where this sort of the field strengths are quite high in the front of the brain and reduced again if you segment the skull accurately. So if you think about a simulation setup where you would use a head model where you have these segmentation errors and then inform your actual stimulation with those kind of head models, you might actually end up stimulating with the wrong intensity and in the wrong place. So that's why we actually want to get this anatomical modeling to be as accurate as possible. So I showed you the die scores, which compare how close the segmentation from the automated segmentation is to the reference we have, which are the semi-automated segmentations. We also wanted to see if the field simulations are sort of similar. So we started out by taking the reference segmentation and then simulating the standard transcranial electric stimulation setup with these 2 pad electrodes. And this is, you can see the field there. And then doing the same using the automated segmentation. And if you only have a T1 weighted scan as an input, we actually get quite big field differences, up to 30% in these fields. And we were a bit surprised about this because the die scores were quite good. But what happens here is that these T1 weighted images are optimized for SNR in the brain and also contrast in the brain, and not so much for structures outside of the brain. So here we have a fat shift. So the bright rim you can see like right above the skull is actually shifted down. It should be higher. And what happens is that the automated segmentation method segments the skull. That's the borders, the tissue borders from the automated segmentations are in this yellowish color, and from the reference in red. So the automated segmentation approach actually segments the skull a bit too thin and the skin too thick. And this is enough to actually cause these quite large field differences. If you include a T2 weighted scan, the die score didn't go up so much, but now the field differences go away actually mostly. And here's the reason is that we actually get extra information with the T2 weighted scan. The CSF lights up. We can pick out the CSF bone border better. We don't have this fat shift issue so much, and we get a more accurate segmentation of the skull and the scalp. So that's why we now, when you do these individual simulations, we recommend that you also have a T2 weighted scan to get these sort of segmentations as accurate as possible. just to show that it's mostly driven by fat shift, this effect, we also have these M-Dixon scans where the fat shift is controlled. And here we get more accurate segmentations again of the skull and the scalp, and the differences are much smaller. If you do the same thing for TMS, the differences are not as big, so it's in the -10 to 10% range, but you see the same trend. If you just use the T1 weighted scan, differences are higher, include a T2, and they go down. And also if you use the sort of Dixon sequence, you get more accurate segmentation, and then these field differences also go down. So now we have these segmentations with 14 different tissue classes, and typically what is used in simulations is 5 different tissues. So we wanted to see how much does it affect the fields if you actually include more tissues in the simulation. And here's an example of an electric stimulation and magnetic stimulation. And the first field distribution you see is from this extended head model where we now have 14 different tissue classes. And the second one is from the standard head model with 5 different tissue classes. And the difference is just a node-wise difference in the fields or the brain or the cortex. So if you look at the difference map, what you see, for example, is these field peaks on top of the brain in the cortex. And this is because we now have the vein on top of the head in the model. And that doesn't conduct electricity as well as CSF. So what happens is actually the current shunts through under the vein. And you get these field peaks in the cortex. You also get reduction of the field in front of the brain, and this is because now in the extended model we have compact bone, spongy bone separated. Compact bone has a lower conductivity, and for this particular subject, the front of the head was made mostly out of compact bone, so we get this field reduction there. For TMS, we chose sort of this location of stimulation so that we can show the vein effect. But if you would do, for example, pre-SMA stimulation, and the coil would be sort of placed on top of the head, and the currents are flowing from left to right here. You can see again we have these field peaks in the cortex, and that's because the current again shunts under the vein, and you get these sort of field peaks there. Again, these sort of differences are in the 10% range. The caveat is, of course, that we don't know the tissue conductivities really well, and that's actually a good segue to the next part of the talk, because Guilherme is going to talk about how to quantify the uncertainties in the tissue conductivities and map them to the fields.
Hello, good afternoon. So I'm Guilherme, and I'm going to continue the talk now, talk about some things that you can do after we have the models done. So first I'm going to talk about uncertainty quantification, which is about how to deal with uncertainties and how to incorporate those in the model. And then a little bit about TES electrode optimization. So conductivity values are very invasive to measure. Normally they are measured by invasive insertion of electrodes. So it's not something that we can do in a subject by subject basis, of course, and it's not something that is actually done very often. We have to rely on literature values to get the conductivities, and the literature on the topic can be very sparse, and the values in the literature can also have a quite large range reported. So it can be very difficult to figure out what would be a good conductivity value for any given tissue. And we want to know, okay, how does that affect our final estimates and how can we incorporate that? So to do it, we set up uncertainty quantification and sensitivity analysis algorithm based on the generalized polynomial chaos expansion. This is, the method is based on sampling from what we do is that we represent the uncertainty that we have in the input, in the conductivity distribution as a probability density. So as a probability distribution, we sample from these probability distributions and get electric fields out. And we use a polynomial, basically, a polynomial expansion to go from the conductivity values to the output distributions, how we chose these polynomials and so on. There's so it's a lot of work goes into how to choose these polynomials and how to do this expansion carefully, and we have a good deal of our paper is about that. And what we have afterwards is that we have a model for E as a function of the conductivity values. And we can use this model also to go back into the sensitivity analysis, so we can say how much of the uncertainty in the output can be attributed to individual input conductivities. Now some results. So it means that after this analysis, what we have is actually, instead of having a dielectric field just as one value of dielectric field, we have a electric field distribution for each point in the head, and we can also go back and attribute this, look how each of the input, how each of the input conductivity uncertainties affects the output distribution. So here, for example, we have 3 different stimulations, 1 TMS and 2 TES stimulations. And we show the mean electric field, so what the expected value from the uncertainty, and the relative standard deviation. What we see, first of all, is that the TMS is far less affected from the tissue conductivity uncertainties than the TES. And that what we would expect, because the TMS has, because the basically how they generate the fields, TMS is far less prone to pick up this, is far less vulnerable to the conductivity distributions. And also it's interesting to note that how this distribution here looks like. So what we have is a far more uncertain field in the SOCI than in the gyri, especially for the TMS case. It's also seen here for the standard TES case, but it's not so, maybe not so true for the focal one, where we also have a higher uncertainty in the gyro crowns. However, the field here is also a lot smaller, so we also need to take that into account. So it's a larger variation on a smaller field in the first range, in the first place. So what we can do, so in the sensitivity analysis, for example, we can do is to calculate this Sobol coefficients. The Sobol coefficients, what they do is that we get the variance of the whole distribution of, for example, the electric field in each at each position, and we can decompose this variance into different components which can be attributed to individual tissues. So, for example, here is the variance that's caused by the white matter conductivity uncertainty, by the gray matter conductivity uncertainty CSF, CSF, spongy bone, compact bone, and skin. We see here, for example, that the gray matter is a very important factor for all of them. So if we were able to measure one conductivity value, maybe put efforts into measuring one conductivity value and try to get it, get a better estimate, or we should probably try to measure gray matter conductivity better. But also that the TES is also very dependent on the compact bone and on the skin conductivity uncertainty, which is also expected because, sorry?
Where are the columns?
The columns? So the columns are the 3, these ones. So it's the different simulation setups. So this here is the focal, this here is the TMS, this here is the focal TES, and this here is the standard TES with 2 pads. And yeah, so yeah, we can see here the TMS one, the variance is quite small, so it's hard to see anything from here, but the gray matter one might be a little bit more visible, while for the other ones it's pretty clear that the gray matter, compact bone, and skin are the major factors for causing the uncertainty in the output field. And one very nice thing about this framework is that it's very flexible. We can expand the electric fields, but we can also expand basically any other quantity we want. So here, for example, we expanded the maximum electric field using the same method, the maximum being measured as a 98th percentile. And we get, as we see, an output distribution for the maximum electric field, which is, so maximum. And again, we see TMS is far less uncertain, so the distribution is a lot narrower than the TDCS ones shown here below. And we can also calculate the same thing, so the Sobol indices, which is a decomposition of the variance. And we see again that the maximum is more vulnerable to the gray matter, conductivity uncertain, and for TMS also white matter is an important component, and for the DCS, compact bone and skin are the other two most dominant factors influencing, in this case, the maximum electric field. And we also look into how uncertain is the electric field pattern. So instead of looking at the total of the maximum field, we want to see how, if the where it's being simulated changes. So how much certainty can you say about where the maximum is independently of what the maximum actually is? And so we use this RDM measure to try to measure the pattern differences from one reference value, which is the mean from the input. And what we see is that it actually does not change so much. So it means that actually the electric field pattern is fairly stable. However, as we saw previously, the maximum, so the absolute values vary quite a bit, which is, I think, an interesting finding. So we can at least rely on this here more or less, as far as we trust the segmentations, which were not tackled in this study. Okay, now I'm moving on to another topic, which is TES electrode optimization. So what we want to do is we want to use the modeling to automatically optimize electrode placement, to automatically figure out where we want to place electrodes, TES electrodes, to simulate a particular target. And I mean, this also transfer. This is also going to be quite easy to transfer to the TMS case that you guys working here. So first of all, what is optimization from a mathematical perspective? So in optimization, we normally have an optimization problem normally looks like this here. So what we have is an optimization variable X. In this case, it would be electrode positions and currents. We have an objective function X, F0, that we want to either minimize or maximize. Could be some measure of electrical focality or intensity. And we also have constraint functions. We can have many constraint functions which can set, for example, limits to currents or number of available electrodes. There has been quite a bit of previous work done in this area, highlighting especially the work by Dmowski, which is the first one to really try to do a more practical application of optimization into TES. But of this previous work differ in terms of definitions that they use for objectives, they use different constraints, and they use different solution methods. So we thought that there is still some room that we can work with and that we can still try to improve it. What they mostly have in common is that they use this formulation called, most of them use a Leadfield Matrix. So because the electric field E is linear with respect to the injected currents, if we consider a fixed grid of electrodes, we can calculate a Leadfield matrix A such that for any electrode current combination X, we can calculate electric field E. Now this matrix here is quite large. It has something of the order of 10 million entries. So we must be careful into how we handle it. But it's a very practical approach where we can combine. Once we have this matrix done, we can easily combine we can easily try any sort of electrode combination. Now, the definition that we mostly worked with is this one. So what we try to minimize is an energy term, which is proportional to the square of the electric field. Now, luckily, because of this proportion to the square of the electric field, and also by design, we can basically collapse the big dimension on this Leadfield matrix. So we end up with a cube matrix, which is just the number of electrodes, which is of the order of a few hundred or even less than that, which makes the problem a lot more manageable. And so what we wanted to minimize is field energy while we match a desired average field in the target. We also have to follow Kirchhoff's law, so different formulations because we need to inject the same amount of electrode that we take out. We also have constraints to total current injected and constraints to current injected through individual electrodes. We like a lot this formulation because it has relatively few input parameters. So it has a target position and a target direction, which are implicit here in this L matrix. Now it has, we have to specify a target, a field that we want in the target position and direction, which is this T, and we have to specify current limits. There is no weights or no regularization, nothing like that is involved. So the problem is using basically only physical parameters. And this here turns out to be a quadratic programming or QP problem, which can be very efficiently solved. So in our implementation, we solve it in around 0.7 seconds for 300 electrodes. So time is really not an issue there. So let's see how it looks like. So in practice, so Oula has shown this picture before. If we have, for example, bipolar montage, standard montage for motor cortex stimulation, or place one electrode over the suborbitally, another one around C3. We have an electric field in the target that looks like this here. So in the motor cortex, that looks like this here. But we also have stimulation of a very large section in the frontal regions. Now we can optimize the montage to produce the exact same field in the target, and the resulting electric field looks like this. So it's a lot more concentrated around the motor region. And the good thing about this here is specificity. So if you observe some effect from the in our study, it's a lot easier to attribute it to specifically motor cortex stimulation if your field looks like this, rather than if it looks like this. Yeah, and of course we have some parameters that we use. So here we use 200 electrodes, 2 milliamps total current, and maximum 1 milliamp to each electrode. Now, if we have a target that's located deeper, so for example, here around the pre-SMA, we can try to stimulate it by placing electrodes bilaterally, which causes a spread out electric field, or we can use an optimized montage. In this case, it doesn't really work out so well. We still have a large section of the cortex which is stimulated, which is quite pretty far away from the target region. And so what this example shows is that the electric field can be highly dependent on the target position. So what you get from the optimization procedure, just because of the physics, because the current has to flow through somewhere. So in this case, it has to flow through more through the gyri in order to get to the deeper brain regions. And that causes the electric field in the gyri to actually be quite large. So what we did with this in mind is to, because the optimization is so efficient, we can just do a mapping study. So we got 20,000 target positions all over the cortex, and we optimized the electric fields for each target position individually. The target was placed normal to the cortex. The target direction was normal to the cortex, and the target intensity was 0.2 volts per meter. And what we see here is more or less what in the left is that the targeting error is basically how far away the maximum is from the actual target, or in the right is this effective area is a focality metric, how large, the larger the worse the focality is. What you see is more or less what you would expect, that if we can get a lot more focal fields in the gyrocrowns rather than the gyri, rather than the sulci. But there is also some more or less unexpected things. For example, here that the field in the in superior regions is can actually not be focused as well as more lateral regions. That might be because of the amount of CSF, for example. If we have more CSF, it might be harder to focus than if we have less CSF because the current just spreads out more. And another thing that we did not look at until now and was limiting the number of electrodes. So previously we simulated 300 electrodes, and in theory, all of them could be active in an optimization result. And we want to, but in practice, we want to limit the number of electrodes to a small number n. Now, this constraint takes this form, which is an L0 or cardinality constraint, and it's combinatorial, which means that it's quite hard to incorporate that into the optimization and still into the optimization procedure. Simple procedures to try to do it are suboptimal, and we know because we tried those for comparisons. And there has been previous attempts to tackle this issue, but they all reported to take a few hours to converge, so the mapping would not be feasible using these kinds of methods. So we designed a branch and bound method. This branch and bound is based on, so we have a state space, which is all the electrode, all the possible electrode combinations, and we subdivide this state space and calculate upper and lower bounds for the optimum values. So for the what would be this energy value, the optimal energy value, and this method converges to a global and certified optimum, which means that we can get that we avoid local minima, so we converge, and we also get a certificate saying that we are within a certain range of the optimal value, which is really good. And for this to work well, this upper lower bound need to be as tight as possible, so they need to be as close to the actual optimal value as possible. For that, we use a combination of L1 constraints to approximate the L0 constraint. And it should allow the state space also be carefully defined to allow for fast pruning. So we want to quickly eliminate places where we know that the solution is not. So with this, our implementation of the branch and bound method with also like our designed upper and lower bound functions, we get converged in around 30 seconds. And if we have some additional heuristics, we can go to around 1.4 seconds. For optimizing with this constraint on number of electrodes. So we now can answer the question, how many electrodes do we actually need to get a focus stimulation that still has some reasonable intensity at the target. So here, what we did the mapping again with the 0.2 volts per meter with a target of 0.2 volts per meter pointing normal to the target. With n equals 4, it's 6 and 8 electrodes maximum, and we calculate difference in the focality metric in relation to the unconstrained case. So we see that with n equals 4, we get worse focality in most places. So it would be better to use more than 4 electrodes, at least under this specific constraints. But if we use 6 or 8 electrodes, the losses are quite small, and they are very much in specific regions. So it means that with the take-home message here being that probably it's not worth buying a system with a TES system with, for example, 200 channels, because you cannot really benefit from it. The most benefit that you get is only of around 8 channels. And yeah, but these findings are also related to the safety constraints that you used. So namely, if we allow for more current in, we can use more electrodes. Or if we allow for less current to each electrode, we can also probably make use of more electrodes. Same if we also want a smaller field at the target, we can also make use of more electrodes. Yeah, so for a conclusion, we are developing SimNIBS, which is a tool for creating realistic and visualized head models and simulating electric fields, and also optimizing electric fields. We have Charm that's going to be included for robust head modeling. And we saw some examples where we scan parameters and which scans you input matter for having accurate head models. And we have errors from segmentation, numerical, and conductivity values. So the numerical aspect I did not cover today. And what we need to do is to quantify them and manage them, and try to minimize them in the future. And what we have in the optimization methods can inform us how to best place TES electrodes and what you can expect from a focal stimulation. Yeah, so I'd like to thank our colleagues and our collaborators. Yeah, and you can visit simnibs.org to get the software. Thank you.